I'd rather just do like 45 push-ups.
And I go, and then I go back down. Yeah, I'm thinking about next week. Like, in this situation, I should probably be able to pull up my body. I actually think about that. I think about that's my brother, actually.
I would die. I'd like to. I'd probably just like that. It's inevitable.
Like, that's what I'm saying, is I think about that all the time.
No, like,
I would,
I would like, get to the other time, I would say that my legs are holding me, and I would just be hanging upside down.
I just think about like the time, like, if I was at a divergent.
I need to watch that. It's so good.
I think I'm saying here. Saturday, Sunday.
Just like that's what I want to say.
I saw I was gonna go on Friday, and there was like broccoli trade, and I was like,
Yeah, you actually can't leave.
I'm gonna run just to my office and grab the attendance sheet real quick.
Because we need service that we're just gonna die semester, and that'll make it less.
Oh, you're allergic?
That'll make me worse next time. I'm just not breathing.
No, that's my neighbors. Down the hall.
Yeah, there's always a dog on the first time.
The other one came over too.
One time I was walking to the room, and someone opened the door and the dog ran in, like they let the dog up, and I was just like standing there, I was like, this is the best day ever.
Oh, yeah. I was doing dishes and I had my noise kids like headphones on, and one of the dogs in the dorm like jumped on my leg and it scared. Like he literally jumped and yelled. I was like,
I one day, like, not help there, but just like in there. She loves to sit on her couch, like, I'll let her in my room and just let her go, but she immediately, she goes, to the next room on the couch. It's like her, like,
you all have the same schedule.
All right, good morning, everyone. I'm gonna start by just taking attendance. Tegan? Lucia? Nick, or Nicole? Autumn? Ryan? Quinn? Celeste? Zoe? Here. Annika? Here. Jesse? Sarah? Here. Kata? Clayton?
Here.
Paige? Erica?
Here.
Victoria?
Here.
Brooke? Abby?
Here.
Hannah?
Here.
Gabby?
Here.
Juliana? Nick? Henry?
Here.
Tyler?
Here.
Everyone's here. Cool. So today we're gonna talk about some things in chapter 4. And so we're going to talk about some bacterial culture, growth and development, how bacteria grow and develop, and how we culture them. So this will be the last content material on your exam next week. And so I have another worksheet that you guys can do. This is on lectures 4 and 5. And so that's the end of chapter 3, and then today, chapter 4. So we'll review this on Monday, and we'll have like a review day. If you guys have questions, you can ask. And so we'll kind of do it like that. So yeah, I'll pass this out at the end, or before you leave, grab one, and then yeah, fill this out, and we can talk about it on Monday. So today, the suggested readings of the chapter are up here, just 4 sections in chapter 4. So we're going to talk about how microbes take up nutrients, how they're cultured and counted, and about some biofilms. So Like all things, microbes need carbon and energy to grow. They've evolved to find, acquire, metabolize a wide assortment of different resources for food. And we know this because we know the microbial genomes, all the genes that microbes carry, have a lot of plasticity, which just means that they're able to turn them on and off and adapt to different environments. And so when resources deplete for bacteria, there are 4 things that can happen, and they're in the order, the order that they're in is what's most common to least common. So when resources deplete in the environment, bacteria will most of the time die. In other situations, bacteria live in communities with other bacteria. So in some other situations, they can eat the other bacteria and basically cannibalize unfortunate others. And then the third thing that can happen is that they can kind of hibernate or like hunker down and wait for nutrients, or they can search for nutrients. And so they can either hunker down and wait by forming spores. So we'll talk a little bit more about spores later on, but essentially they are these structures that bacteria and fungi can form so that they can kind of like hibernate, hunker down, become latent, and just stay happy and survive, but then they're there waiting for nutrients. Or they can form flagella, and so they can have these motility devices. So flagella are one of them. So that allows for searching for nutrients. And then lastly, this very rarely happens, but there can be mutations that occur, and so that allows the bacteria to evolve and to use different or remaining resources a little bit better. So this is more rare. It really has to have perfect mutation occur for it to be able to adapt in that way, but it does happen, and that's part of evolution. Okay, I'm gonna just close this door a little bit. All right. So yeah, there's bacteria, like I said, they grow really, really fast. And so one of the things that I just kind of want to drive home is that there is a finite supply of nutrients. And so yeah, they grow really, really fast, but they go from feast to famine really quickly. And so they always, in the environment that they live in, they're always like feasting, they use up all the nutrients, and then once those nutrients deplete, then they need something else, and then they go into famine mode, and then these are some of the things that can happen. So just wanted to make that clear. So despite them being very plastic and being able to adapt and things like that, we briefly talked about this, but most bacterial species fail to grow in lab. So this is just a fun schematic that was found on what was formerly Twitter, X. And so bacteria in nature literally can like eat dirt. They defy the physical limits of light. They can exceed, like, they can live in like these most extreme temperatures and environments. And so like here, there's this bacteria that's like super strong, and it's the third eukaryotic extinction event in a row, and they've still been around because they're prokaryotes. But then bacteria in the lab are just really, it's really amazing how difficult it is to grow these suckers. It's really hard to figure out the exact secret sauce to grow certain things. So bacteria in the lab, like you give them a sugar and they're like, nah, I don't like lactose, and so then like they won't grow. Or if the pH is off just by a tiny, tiny bit, then they're like not happy and they don't grow. Or literally they're like, oh, this is tap water? No, thank you. So like they're just very picky, and I just really like to use this analogy because that's kind of, you know, how it works, and it's hard to figure out the exact sauce of how to grow them in the lab setting and replicate the environment that they live in. All right, so how do microbes build biomass? We're going to talk about a little bit about this. I'm not going to go into too much detail on this stuff because it is important for microbiology, but it also overlaps with some other fields of biology. So I'm going to just talk about the most important things. And one of those things is carbon. All of Earth's life forms are based on carbon. Like I said, everything, including microbes, need carbon to grow. And carbon can be acquired in 2 different ways. And so there's this cycle here that I'm going to tell you. This is the most basic form of the carbon cycle, where you have these autotrophs that fix carbon dioxide. So autotrophs fix carbon dioxide, and then they take that carbon dioxide and they make organic molecules like sugars. So autotrophs use carbon dioxide and make these glucose molecules or organic compounds, and then those compounds are then taken up by heterotrophs, who are able to use the organic molecules to then generate carbon dioxide for autotrophs to use, and the cycle continues. So we need both autotrophs and heterotrophs. So these are the two trophs that I think you guys should know. There's so many others, but we're not going to get into it in this class. So the carbon cycle really runs on autotrophs and heterotrophs, and so this is the more I guess, advanced version of this. You don't have to remember everything in here, but I want you, what I want from you to take away is, so on the left we have this heterotrophic metabolism, and so heterotrophs gain energy from degrading polysaccharides, so those are the organic compounds like glucose, and they break it down into smaller compounds, like here we see the 6-carbon glucose, and then the glucose will undergo glycolysis, where it makes 3 carbon pyruvates, 2 molecules of pyruvate. And then pyruvate normally will move through the TCA or the Krebs cycle. And during this process, a lot of carbon dioxide is released. In the absence of this TCA cycle, however, the carbon can end up as fermentation products. So acetate is one of those fermentation products that we'll talk about later. So pyruvate goes through the TCA cycle, and it can either produce a lot of CO2 or become turn into acetate, which is then also gets produced as CO2. But what's important here is that this CO2 is then used by autotrophs. And so autotrophs use energy from either absorbing light or the oxidation of minerals. So that's phototrophy or lithotrophy. We're not going to go into that. You don't have to know these words, but essentially you should know that autotrophs use either light or mineral oxidation to process that CO2. And so this energy is used to convert or fix CO2 into these complex organic molecules like glucose, and then the process repeats, and heterotrophs use that glucose up. So what's important here for you to remember is that CO2, or carbon dioxide, is made from heterotrophs, and then it's used as the inorganic carbon source for autotrophs to make organic compounds like glucose, and then the cycle continues. Does that make sense? Is there any questions at all on this? Okay. So the carbon cycle runs on this cycle between autotrophs and heterotrophs. But then what about nitrogen? So nitrogen is the most abundant gas in the Earth's atmosphere. It makes up about 80% of the atmosphere. But what's interesting is that it's unavailable for use by most organisms. So nitrogen gas is N2, and the way that the chemical bonds are, or the way that it's structured, is you have these 2 nitrogen molecules that are bonded by this triple bond. This triple bond is really, really stable. It's super stable between these nitrogen atoms, and so that's the reason that we can't use nitrogen gas from the atmosphere ourselves, and the majority of organisms can't use it either. So what needs to happen is there needs to be certain microbes that can fix this nitrogen gas into something that we can use. And so essentially what happens is that it's fixed into these ammonium ions, so NH4+, that's ammonium. And so the way that this conversion is done is by certain bacteria, like rhizobium here. These are called nitrogen fixers. And so they basically fix the nitrogen into a usable ammonium. And these nitrogen fixers are important because the way that they do this is they possess an enzyme called nitrogenase. And that nitrogenase, remember, anything that ends in ase is like an enzyme. So it's an enzyme that they use to fix the nitrogen and convert nitrogen gas into ammonium ions. And then these ammonium ions can then be used by all microbes. So there's specific microbes that can fix nitrogen, but then there's these other microbes, majority of others that are nitrifiers, that can use these ammonium ions to then make their own cellular components. And then these can also be used by plants. So these nitrifiers then oxidize the ammonium or convert the ammonium to nitrate, which is shown here in NO3. And then finally, we have these denitrifiers that can convert nitrate back to nitrogen gas. So this is essential for all of global health. And so these are some of the key things for you guys to remember here. Any questions on this? All right. I'm going to show you a cool, hopefully this works, little video about nitrogen-fixing bacteria that have been found on corn in Mexico.
Corn. It's the dominant crop of the developed world, a highly valuable commodity used to make food and fuel. But in order to grow, corn needs nitrogen. And although nitrogen makes up 78% of our atmosphere, it's not in a form that corn can use. So, like most commercial crops, corn, or maize, gets its nitrogen from synthetic fertilizer. Synthetic nitrogen fertilizer has helped to feed billions of people, preventing massive global starvation. But it comes at a cost. Over $80 billion a year globally, over $11 billion in the US alone. 1 to 2% of the Earth's energy supply is used to make the fertilizer, creating 1 to 2% of the world's carbon footprint. Then even more energy is used to transport and apply it. And much of the nitrogen escapes in the form of greenhouse gases. And fertilizer also runs off the fields, ending up in our waterways, causing an enormous dead zone in the Gulf of Mexico. It's a huge problem, and it's getting worse every year. But in the mountains of Mexico, there's an indigenous variety of corn grown in an isolated community for centuries near where corn first originated. A unique trait appears to have evolved in this type of corn: nitrogen fixation.
This corn appears to be able to fix the nitrogen from the atmosphere into a usable form.
Biological nitrogen fixation describes a plant's ability to pull in and use nitrogen from the atmosphere instead of requiring fertilizer. This occurs when an enzyme called nitrogenase converts atmospheric nitrogen into ammonia, which can be used by a plant for growth. This cutting-edge research has its roots decades ago, when Howard Yana Shapiro, who's now Mars's chief agricultural officer, observed this unique corn in a small community in Mexico.
Just didn't have the next generation equipment, the next sequencers. We didn't have a way to analyze this, and the whole notion of microbiomes or metagenomics wasn't even invented. So it's great to live long enough to have the science catch up to answer a question that you had.
This corn generates a series of aerial roots. Turns out this is an extremely unique morphological trait.
The indigenous corn produces large amounts of mucilage from aerial roots between 3 to 6 months after planting.
During certain times of the year, there's a secretion of mucilage or mucigel, which you can physically see dripping off the roots.
This mucilage provides a low oxygen and sugar-rich environment for the diverse microbes. This serves as a site for nitrogen fixation.
The mucilage provides a unique home or environment for these bacteria that fix nitrogen. They can restrict the diffusion of oxygen, and oxygen kills the ability of bacteria to fix nitrogen. So the mucilage restricts oxygen, creates a low oxygen environment, and it provides sugars. It's feeding the bacteria. So classic symbiosis.
It was It's really the wisdom of the Mars family that made that possible. Through direct funding to the University of California, Davis, they understood what would happen if you could reduce or eliminate the nitrogen applied to agricultural fields around the world.
All right. So, yeah, I hope that what you took from that is, we've eventually now have evolved to have all these techniques that allow us to recognize in these certain plants nitrogen fixers that fix nitrogen gas. It needs to be in an anaerobic environment, which means very low or no oxygen. And so there's been this evolution where those plants form that mucin gel that basically coats everything around it, and it provides this like space for the bacteria to grow and be able to fix nitrogen. And so that's just really cool to see. So that's really all we're going to talk about in terms of cycles and how microbes, take up their biomass. And so now we're going to talk a little bit more about the bacterial cell wall again, and so the membrane and how nutrients get in and out for the cell. Any questions though on the carbon and nitrogen cycle before we move on? Okay, so like I've said already, membranes separate what's outside the cell from what's inside the cell. And selective permeability, so selecting what's able to get in or out of the cell, is achieved in 3 different ways. The first one is you can have specific substrate carrier proteins called permeases that basically permeate the membrane. Or you could have dedicated nutrient binding proteins that patrol the periplasmic space. You can have membrane spanning protein channels or pores. So this is more of a memorization slide for you guys, just to know these 3 ways. And so now we're going to talk a little bit more about a couple of different kinds of transport across membranes. The first being passive transport, so facilitative diffusion. And so in facilitative diffusion, it helps solutes move across the membrane from a region of high to low concentration. So that is when solutes are moving not against their concentration gradient, they're moving towards it or up it. In passive transport or facilitated diffusion, it does not use energy, and then it only allows compounds to diffuse across the membrane that are going basically towards its concentration gradient from high to low concentration. I'm going to show you an example here of facilitated diffusion. So this is a glycerol transporter of E. coli. So over here you have this transporter protein that's embedded in the membrane. And so this is called GLPF, so it's a glycerol-specific transporter. It basically transports glycerol, facilitates the diffusion of glycerol into the bacterial cell. So this is found in E. coli. And so here the glycerol enters this channel. And then as soon as glycerol is bound inside of that channel membrane, that activates a conformational change in the GLPF in this transporter that opens the channel to the cytoplasmic side, and glycerol enters the cell. So over here you can see that there is a higher concentration of glycerol. There's 4 here and there's 0 here. So it always is going to go from high to low concentration in passive transport. So it's important to know that this can reversibly and randomly adopt one of the 2 conformations. So the first form exposes the glycerol binding site to this extracellular space, or it could be in the second form where this it's exposing its binding site where it's going to release the glycerol to the cytoplasm. And one of the things about this is someone might think like, okay, what if you have all 4 of these come in? Now you have a higher concentration of glycerol here. How is this going to stay as a passive transport type of mechanism? The reason is because in this specific one, in this mechanism, glycerol, as soon as it gets into the cytoplasm, it gets used up. And so as soon as it's coming in, it's getting used up. So it's always going to be this case of it going through from high concentration to low concentration. So it's going to be facilitated diffusion. So it keeps the concentration low inside of the cell. Yeah.
Is that second bullet point saying that compounds that are too large or too polar can't diffuse, or they can?
Yeah, that's a good question. I just realized the wording on that is it's difficult. So yeah, they can't diffuse across the membrane. Yeah, so it only, because if you have a polar molecule, it's going to be charged, and so it's going to need like active transport. Yeah, good point. Thank you. Anything else on this? I hope that makes sense. Now we're gonna talk a little bit about active transport, and this requires energy. And so this is when things are moving against their concentration gradient. So you're going from low concentration to high concentration. So that's not natural, and so it needs energy. And there are two types that I want to talk about that are coupled transport systems. So there's transport systems that are coupled. And basically, energy that's released by a driving ion moves down its gradient naturally. So the driving ion is moving via facilitated diffusion. But then that energy that's released from that is used to move a solute up its gradient. And so that's the active transport. So the first kind that I'm going to talk about is symport, where two molecules travel in the same direction. So here, we're looking at red molecules and we're looking at blue molecules. And so the first substrate, so we're going to call this the driving ion in red. So there's a higher concentration up here outside of the cell versus there's just 3 of those inside of the cell. And so that's going to naturally want to go from high concentration to low concentration and facilitated diffusion. And so when that goes through, that's going to create energy that's released, and this energy is able to then move a second substrate, so the blue one that's low concentration outside of the cell will move against its concentration gradient into the cell where there's higher concentration. So the key thing here is that it's a coupled transport system where energy is released from a driving ion that's moving down its gradient or not against its gradient, so and then that energy is moving a second substrate that's going through this active transport because they are not, because there's a less concentration on the outside of the cell. So this is the simplest way to use energy to move molecules across the membrane, and that's by exchanging energy of one chemical gradient for that of another. And the key thing here in symport is the two molecules are traveling in the same direction. The other kind is antiport. So it's just the antithesis of symport, meaning an actively transported molecule moves in the direction opposite to the driving ion. So it's the same exact concept where you have a higher concentration of the red now here on the bottom, and that is going to move from high to low naturally. So they just kind of switched it. Instead of going from top to bottom, it's going from the inside of the cell to the outside. So this substrate A here in red is on the cytoplasmic side of the membrane. It's going to naturally want to go from high to low concentration, and then it opens up on the outside when it releases this ion. And once it does this, where the concentration of this red molecule is less, it's going to now leave its binding site, and then there's another binding site for substrate B in blue to bind into that site and then move against its concentration gradient from low to high concentration. And then it gets released on the other side, and then you have the same thing here where you have higher concentration of the red molecule going back in and getting out. And so it's the same thing where the first molecule is not going against its concentration gradient, but the energy used from that is moving the other molecule against its concentration gradient, because that will require energy. But the difference between antiport and symport is that the antiport means that the second molecule is moving in the opposite direction of the first molecule. Does that make sense? Okay. So yeah, both function in the same way by opening one end or the other of the channel. So that's not where the difference is, like what side of the channel it's opening on. It has to do with going up or down this concentration gradient and the energy being produced, and it's just different directions that the second molecule is moving in. All right. And so the last thing we're going to talk about transporter-wise is these ABC transporters. These are ATP binding cassette transporters, and so that's why we call it ABC. And they're the largest family of energy-driven transport systems, and so these are important because they are present in all three domains of life. So you can find these in really like many different organisms. It's really highly utilized, and it helps really drive a lot of energy to transport things. So there's two main types of this. There's uptake and efflux. So we'll talk a little bit about that. But first we're going to talk about it in the context of it being uptake. So here, these are really important. So here we have, this is this a gram-negative or gram-positive bacteria? Let me ask you that. You see an outer membrane and an inner membrane. Negative. Gram-negative, remember, gram-negative has the two membranes because it has the really thin peptidoglycan layer, which is not shown here. But the gram-positives have that thick peptidoglycan layer, so they will only have one membrane. So this would have been a gram-positive cell that they're showing in this schematic. Okay? Not to say that this doesn't exist in gram-positives, but it's just the way that they're showing it. And I just want to make sure you guys can still tell the difference when you're shown a different type of image. So you have this, what is this? This is a paraplasmic binding protein. So it's just a binding protein, and this is needed in order to bring solutes into the cell. And so you have this porin right here. That's what this, this protein is. So this will bind through, and this solute will come in, and I'm sorry, actually, this solute binding protein is in blue here, and in yellow we have the porin. I misspoke there. So the porin brings in the solute that binds to this protein that's supposed to bind it, and then this protein brings it into this membrane transporter here shown in green. And so once this happens, the solute is able to bind. Sorry, my mouse is moving weird. It binds into this membrane transporter, and once it binds, it activates a conformational change. So once it binds, you have this side open up, this side closed, and then this ATP binding cassette shown over here dimerizes once that conformational change happens, meaning that it becomes a dimer. It becomes two molecules fused into one, and that's when it becomes active. So once this happens, ATP is hydrolyzed, which just means that ATP is converted into ADP plus a phosphate. So ATP here is really important. It powers this whole thing. And then once ATP is hydrolyzed, this dimer then opens up and it releases the solute into the cytoplasm. So this is important. This happens a lot in cases of just taking up really critical nutrients. You have sugars and amino acids the cell needs. So they have these ABC transporters. So you have this porin that brings in this important solute or nutrient that it wants. There's proteins that bring it to the ABC transporter and the membrane protein, and so that all gets activated through conformational changes in ATP hydrolysis, which releases the solute into the cytoplasm. So it's ATP hydrolysis that's opening this channel. So like everything, there's been a lot of evolutionary advances in bacteria where they've also now started to use these ABC transporters as a way to create efflux, which means that they pump out things that they don't want. So basically, some bacteria have evolved to pump out antibiotics that we try to give to kill the bacteria, and they use this system. So that's what efflux means. So it's generally used as a multi-drug efflux pump. Any questions on this? Let's do a quick concept check. So all of the following statements about ABC transporters are true except so let's talk through them. Do they require energy? Yes. Are they found only in prokaryotes? Remember, I said that these are so important and they exist in all three domains of life. So prokaryotes, eukaryotes, archaea. So that one is not true. So that would be the answer, but let's go through the rest. They can be used for import and export. Is that true? Yeah. So they can be used for import because that's, them bringing nutrients in, and export meaning they can pump stuff out of the cell. And so in the efflux example, and they are the largest family of transporters. That is true. So the answer is B, that they're only found in prokaryotes. These are really important and they are found among all domains of life. All right, so if we're good on that, we're going to talk about some fun stuff that's going to apply to some of the things we're doing in lab, and we're going to talk about how we culture and count bacteria. So microbes in nature, they exist in complex, multi-species communities. So here we have a plate and we have all these different types of species growing on it. But for detailed studies in the lab, they are grown in pure culture. So what that means is they are grown by themselves, so either on a plate or in the broth. We try to get a pure culture. So yesterday in lab, our cultures were not pure because they were contaminated, cross-contaminated with each other. But we tried. So after about 120 years, we've succeeded in culturing only less than 1% of the microbes around us. So we literally like I said previously, is we aren't able to grow most of these in the laboratory setting. And so the percentage of what we have cultured is just about 1%, less than. So the vast majority have yet to be cultured. And so this is an exciting time to be a microbiologist if anyone is thinking about it and I haven't convinced you yet. So bacteria are grown in two types of culture media. We've already done one of them in the lab where we have liquid or broth culture. So these are useful for studying the growth characteristics of a pure culture. So we definitely tried to do that in lab yesterday, and we'll do more of that. But then there's also solid media, which you'll play around with a little bit more, and liquid cultures next week in lab. So solid media is useful for trying to separate mixed cultures from clinical specimens or natural environments. So here we have a handprint, and so all the different bacteria are on here, and you can really distinguish differences in colony types. This is a fungus over here, because of how large it is first, and then all of these hyphae that are starting to form. And you have all these different types of bacteria that grow in different sizes and different shapes and different colors. So putting things on a plate helps you to actually isolate individual colonies that are contributing to that one species. So in this one colony, this is composed of millions of bacteria, but they are all genetically identical. So that's how we know that it's pure. If we want to we can pick this colony and put it in some nutrient broth and then just grow just that up. So that's one way to then isolate one, and so this is useful for studying the growth characteristics of a pure culture. So you can't see mixed cultures in a liquid broth, but you can see them on a plate. So that's why we tend to separate mixed cultures on solid media. And so there are different ways to isolate colonies. So we're going to talk a little bit about how we try to isolate colonies. So here you have a bunch that are also on top of each other. And so these aren't necessarily separated. This one is. But in order to actually get separation from this mixed bag of microbes, we have two techniques. We can either do a dilution streak plate or a spread plate. So you guys will be doing this in the lab, so I will talk about it now a little bit, and then I'll also talk about it again in lab. But when you're doing a streak dilution or dilution streaking, you have a loop. You have your loop that's, you flame it, and then you streak your specimen on in one spot here, And then, so you have this lawn of colonies, which is basically shown by them not being separated, right? So you have this lawn, and then you, instead of going back and dipping back into the bacteria, you basically set the highest concentration that you just picked up over here, and then you flame your loop again to get rid of everything that's on there, and you're diluting it essentially by then dragging out, I need my pointer, which I don't have because this mouse sucks. All right, let's see. So yeah, you drag out a little bit from this really concentrated part, and then you drag it out here, and then you flame your loop to get rid of the bacteria, and then you drag out from here, and then you flame your loop a little, and then you drag out from the third quadrant, and then once you end up going, usually you do it in 4 quadrants, and I'll explain that, you'll then drag out the species onto this part of the plate, and so now when it grows, you see these individual colonies for separation. A spread plate, basically what you're doing is you have a liquid culture, and you do dilutions. Basically, if I have a, let me draw this out. So if I have a tube, and it has this is the amount of broth, and then this is all the bacteria. There's so much in here. And I know that if I put this on a plate, you're not gonna get these individual colonies. It's gonna look all like that. And you're not able to isolate anything. So what then I can do is take another tube and put some nutrient broth in here, some fresh broth, and only take a little bit of this. Say I'll take 10 microliters, which is a very small amount, and I put it in here. So I diluted it, basically. You're diluting the concentration. And then you can do this again and do another 10 microliters. And so you'll have even less. So here we'll do this much bacteria. And then you can put these on a plate, so you can then plate 10 microliters. So you're diluting it again by only plating a very, very small amount. So then you can get individual colonies. So here you'll get, I don't know, several, maybe hundreds, and then here we eventually want to get a countable number of colonies. Does that make sense? Okay. So these are 2 ways. They both start from, well, this one, dilution streaking, you can start from either a plate, or you can start from a liquid culture, but when you're spread plating, you're gonna do these serial dilutions on a liquid culture. So you're doing dilutions in the tube and then you're plating, versus here you're doing the dilution straight on your plate. So dilution streaking, the way that you do it is that's essentially what you'll want to see, but basically, when you're doing a streak plate, you will grab your loop, you'll flame it, and then you'll use your loop, you'll kind of, it's easy to pierce through the auger because it's very thin, but what you'll do is you'll pick up a little bit of your culture from the tube like you've been doing, and then you'll streak in 1 quadrant. I usually actually will have you draw like this as like a quadrant, and I'll label them really small, 1, 2, 3, 4. You have to go in one direction or the other. So I'll basically ignore this part. So this is my first quadrant, and so I'll put all the bacteria here, try not to overlap the other quadrants. So I'll really put that bacteria over here. Then I'll flame my loop, and then I will go into my second quadrant, so my second dilution, and my loop now has no bacteria on it, so I'm only going to grab a little bit of the bacteria, and I'm going to streak it all out over here, not overlapping a bunch of times, because then you're bringing too much bacteria. I always say go in once or twice, but then you'll go into the second quadrant. You want to make sure you really utilize the maximum space in that quadrant, but also not overlapping too much. Then you will go from this quadrant 2 into quadrant 3. So this time I'll go twice, and I'll streak out really well over here. And then you'll do the same thing, flame your loop. You have no more bacteria on there, and you'll grab some of this quadrant and we'll streak here. And so ultimately, this is like a perfect result of what you should see, where you have the most bacteria here, and then it gets diluted further and further. And the goal is to see these individual isolated colonies. Does that make sense? Reflaming the loop before changing direction of streaking is the most important step in this entire process. And then there's spread plating, very similar to what I was trying to explain earlier. So here you have an unknown concentration of colony-forming units. That's another word for just the number of bacteria. So CFUs is the number of bacteria. That's the unit you're using. So you have an unknown concentration usually in the big flask, and you do these dilutions where you see it's getting lighter and lighter because it's getting more and more diluted. And so you do several of these dilutions. They're putting 9 mils of the broth, so the sterile broth, and then 1 mil of from each one into the next. So you're just diluting further and further. And then you can put these on plates. And so initially, the first even few dilutions are going to be too many to count or too numerous to count. And then as you move down your dilution, you should have a countable number of cells. And usually I say a countable number is between like 20 and 200. So you're taking these 10-fold dilutions. You're diluting at 1 to 10 each time. And so that allows you then to estimate the viable density of cells that was in that initial liquid culture. So here you have the 10 to the minus 1, that's just the dilution factor, and we'll go through that a little bit more in lab. But so once you've diluted it once and then twice and then 3 times, you have too numerous to count. You can't see individual colonies. Even at 10 to the minus 4, the 4th dilution, you're still also seeing too many. But then once you get to the last 2, you are having like a more countable number. And so all of these dilution factors, these dilutions and these numbers, and then the amount that you plate is also another dilution factor. All of that can be used in an equation to help you work backwards and find out the number of concentration of colony-forming units in the initial unknown. So it's a really easy, it's a really easy equation. Basically, the number of cells per mil, or colony-forming units per mil, in the original culture tube is the number of CFUs, or colony-forming units, which are all these colonies, divided by the dilution factor times the volume plated. So if we were to use this 10 to the minus 5 plate, if I gave you this plate, or a question like this, and I said, there's 107 colonies growing on the plate that we diluted 10 to the minus 5. How would you, let's try to do this equation together. So we've resolved these isolated colonies. So CFU per mil is the goal. That's what we want to know. You always want to have your units. I'm like a stickler about one thing, and that's about units. So you have your CFUs per mil, which is unknown, is the number of CFUs that you are growing on your plate divided by the dilution factor, which you'll start to see me write as DF, times the volume plated. So what are the numbers that we're just putting in here? How many colonies are on that 10 to the minus 5? 107. Divided by the dilution factor, as we said, 10 to the minus 5 times the volume plated. What was the volume plated? on each of those plates. 0.1 mils. So that's this number right here. This is the volume that's plated on each of these, okay? So because what we're trying to get is in mils, we can just put 0.1. But if I gave you something that wasn't in mils, it was in microliters, you would just have to do the conversion factor. So this is 1.1 times 10 to the 8. Is that right? Okay. And CFU per mil. So that is how that equation works. All right, any questions on that? Okay.
So isn't there any type of bacteria that is affected by the density of the colony surrounding? So like meaning if you're growing with less bacteria surrounded, doesn't that affect the actual counting?
Yeah, so this is a pure culture of one thing. So this is one bacteria. So do you mean because you have so many, it might affect the growth of others around it?
Surrounding density.
Yeah, so when you have really so many bacteria, they kind of just pile up on top of each other. And that's why you see this. We call this a lawn. The reason being that there's just too many, so that's when you have a lawn. Once you have space on the plate for certain ones to grow, that's when you start to see individual colonies. So that's why we have to dilute it in order to start to see that lower concentration of cells. Make sense? Don't forget to grab a paper. All right. So there's different types of media that we can use. So I'll briefly talk about this. We have complex media. So this is really rich media. It has everything you need. It has amino acids, peptides, vitamins, sugars, nucleotides. It has everything that the cells need. But because it's so nutrient rich, it's poorly defined, which means that we don't know the exact amounts of everything that is in there. So it provides many of the building blocks for cells to grow. It's one of the easiest ones to just put bacteria in and they grow. It's a rich, complex media, but we just don't know exactly how much of everything is in there. Then we have minimal media, which is kind of the opposite. It contains only the nutrients that are essential for growth of a given microbe. Bacteria can grow very slowly then because we're forcing the organism to synthesize its own essential components. So we're only giving it those really essential nutrients for it to survive, but then it grows really slowly because you put it on minimal media. We're forcing it to really make its own essential components. We're only giving those nutrients that are essential, but through its metabolism, the cell's metabolism, it has to make the other things. Then there's enriched media, which there are specific components that are added to the complex media. So enriched media just is complex media and then some additional components that are added. So the next two are selective and differential media. Selective media favors the growth of one organism of another, so it makes certain that one organism grows and the other one dies, so it kills one and makes sure one grows. And differential media then exploits the differences between two species that grow equally well. So rather than killing off one bacteria, both of them can grow, but you're exploiting differences between them. So it could be like a color change or something like that. So if one organism can use a certain carbon source that's on the plate, it can then turn a different color. And so you can then use that to see the two different ones. So that's the difference between those. So a lot of clinical microbiology utilizes media that is both selective and differential. An example is this McConkey medium. That's just the name of it. You don't have to remember that. But essentially, what it does when it's selective and differential, here, it's selective because only gram-negative bacteria can grow on McConkey plates with lactose as the carbon source, so it's a selective media. So basically, only gram-negative bacteria can grow on here, but it's also differential because gram-negative species certain ones are capable of fermenting lactose. Lactose is the sugar that's in here, and those ones will turn pink. And the non-lactose fermenters will appear as uncolored. So this is both selective and differential, because it selects for only gram-negative, gram-positive bacteria cannot grow on this. And it's differential because it can then differentiate between different gram-negative bacteria, between lactose fermenters and non-lactose fermenters. Does that make sense? All right. So, like I said, some species that are adapted so well to their natural habitat, so we don't know how to grow them in the lab. Some are unculturable, and they can depend on various factors. One of those includes that some bacteria are very social, and they refuse to grow unless they're with their buddies. So microbes in the environment around those bacteria are providing something to that unculturable bacteria, to us, that we haven't figured out yet. So that's one of those important things that I want to really drive home, that sometimes they're unculturable because they live in an environment with other bacteria, and those bacteria provide something for them to be able to survive that we just haven't figured out yet. So, techniques for counting bacteria. We've talked about plating a little bit, and there's many reasons why it's important to know the number of organisms in a sample. Obviously, counting organisms is invisible to the naked eye is impossible, and there's different techniques that either measure a different physical or biochemical aspect of growth. And so the cell density, the amount of cells that is derived from all these techniques, may not agree with one another, meaning that they all have their own strengths and weaknesses. So these are the 5 techniques for counting bacteria that we're going to talk about. We talked about spread plating, so I won't go through that one again. Optical density basically measures growth of bacteria in real time. I have an experiment planned for lab where you're going to do this. Hopefully it will stay on my list of things, but what you have is a bacteria in a tube of liquid medium, and the way that you can detect its growth is by basically how cloudy the medium appears as the cells scatter light. So basically, you have your sample here in a tube, and this is all bacteria, and you have a light source. The light source will pass through this tube, or a cuvette is what we call it. So the light source passes through. The more cloudy it is, the more bacteria there are, meaning the less the light can pass through, and so that is then converted into a number that tells you how dense the cells are. So the more cloudy, the more dense, the less light that passes through, and that converts it into a certain value that tells you how many bacteria are in there. So this is, because you're able to do it in real time, basically you're able to have a sample and grow it, and every like 15 minutes or so, you can take a little bit of that, put it in this cuvette, and then leave the actual culture still growing. So you're taking samples out at different time points and trying to measure how much the optical density is, or the absorbance is. And so basically here at time 0, you have no cells growing, and then you can kind of track the bacterial growth over time and get a nice curve. So it's quick and easy way to approximate the cell density, but like it says here, it's an approximation. The problem here is that cell volume, the length and morphology of the cells can alter the optical density, and then also dead cells also contribute to that cloudiness. So it's not only counting live viable cells. So this one doesn't account, or it also accounts for dead cells. So that's one of those weaknesses of optical density measurements. Hemocytometer is a direct counting of cells. So basically you can put them on a slide that has a grid system within it, and basically you do dilutions like we just kind of I showed you with the tubes. You put a tiny little bit of that specimen over on the slide, and then there's this inherent grid system inside of it. So you put a known volume of your culture on that slide, and then you can look under the microscope, and then you'll be able to see the grid system and all of these cells. And so you're able to then, this is kind of what it looks like. And so knowing the dimensions of the grid system, really we have like 16 of these big circles. So what you can do is count like 8 of them, because there's going to be a lot of cells, and then just multiply by 2, because you're estimating that because you have 16, there should be about an equal-ish amount of cells in each grid. The weakness with this is similar to the OD measurement, where dead and live cells look exactly the same. However, there is staining techniques that help you to distinguish from dead cells and live cells. And so fluorescence microscopy, we talked a little bit about this before. So this is direct counting of the live and dead cells. So using chemical dyes, here the live cells fluoresce green because this dye enters all of the cells, but then dead cells fluoresce red because the propydium dye can only enter the dead cells. So we give different dyes, and this propydium is very commonly used in a lot of techniques and experiments to be able to look at dead cells and not count them. But if we only use this green dye, all of the cells would be green. So that's one way to differentiate. The last one I want to talk about is flow cytometry and cell sorting. It's also called fluorescence activated cell sorter, FACS, but I just call it flow cytometry. So the previous techniques that I showed you require you to isolate a sample and then count and then back calculate to the initial concentration of the cells that you're working with. Flow cytometry is this whole really complicated system that allows you to directly count your cell sample without using microscopy, without anything. So it's an electronic technique. It helps count and separate your populations of your cells according to properties. And those properties are fluorescence and side scatter. So I'm not going to go into the details of how flow cytometry works. I did a presentation on it one time in my life, and I still don't know it all that well. But what I can tell you is that the cells go through this system. You put the cells through the sample, and then they flow through. And then there's this sorting system that sorts the cells based on 2 things, either fluorescent, whether they are live or dead. So it uses fluorescence to help tag live versus dead cells. And then also the size of the cells contributes to something we call side scatter. So here the fluorescence level is what we call forward scatter, and then, which isn't written on here, so you don't have to know it, but the cell size contributes to the side scatter. So these 2 things together allow us to better understand, you know, how many cells are in our sample, and they get sorted and they get collected into these different channels over here. So one of the people, actually a lot of people in my lab use this technique basically to, so a lot of them are studying like the immune system, and they want to know the cells that are being, you know, generated in an immune system once you have a certain, you know, disease or infection. And so they'll take a sample of tissue or blood that has a bunch of cells, and they put it through this machine, and then it tells you like this many Th1 cells, Th2 cells, like just different types of cells. So it helps to sort it for you. It just takes time and is not the easiest thing to do. Requires a lot of extensive training. And this is a loud, but just weird little. All right, so quick concept check. What's the fastest way to measure cell density? So hemocytometer counting chamber, you'd have to make some dilutions and then put it on a slide and then look through a microscope and then individually count each cell. So that one isn't really slow to do. You can do it in a day, but it requires you to do some thinking and some counting, so it will take a little bit of time. Optical density measurements, that one, basically you put a little bit of a sample into a cuvette, you press a button, and it gives you a reading. That one's pretty fast. The streak plate method, you can streak your bacteria, but it's going to be invisible. You need to give it a week, a few days to grow. So that requires a few days. And flow cytometry is complicated and is probably going to be the one that takes you the longest because you need to put all your samples in and then receive the output information and then do a lot of analysis to like bin things. So that one is just really long. So based on that, which one would be the fastest way to measure cell density? B, optical density. What would be the second fastest way? Counting chamber.
Maybe like A and C, is that like a hemocentimeter is like easier because time-wise, like amount of time you need for it?
Yeah, exactly. Rather than putting them on a plate and waiting for them to grow, you can then just make your dilution and then put a certain amount of cells, you know how much you're putting, onto a little slide, and then you can count them because they're in liquid versus growing on a plate. When you're growing things on a plate, it takes a lot longer. All right. Okay, so the last few slides that I'm going to talk about is bacteria exhibiting these group behaviors. So quorum sensing is one of them. It's really, really cool. I know one of the professors here studied quorum sensing at one point in her career. But basically what that means is a group of bacteria collectively respond to environmental changes. And then there is swarming, which means that a group of bacteria move across a surface together. They kind of swarm like this. Then there's biofilms, which are a group of bacteria adhering to each other. They need to stay around a source of nutrients, so they kind of get all jumbled up together, and they attach to their surface as well. All of these three types help bacteria resist environmental stressors. So biofilms, in nature, many bacteria form specialized surface-attached communities called biofilms. And basically, this is made from one or many species that can form on a range of organic or inorganic surfaces. So they form when nutrients are plentiful. And so, yeah, they form these really nice surface attachment communities. And an array of patterns can form here. And it helps to increase the colony's surface area and allows greater access to oxygen for the millions of cells that are making up the biofilm. So that's why they stick together. When nutrients become scarce, individuals then start to detach and forage for new resources. So like I said, bacteria go from feast to famine. So biofilms will then break apart and then come back together depending on the nutrients. Here's another way that biofilms can exist as plaque on your teeth. So when you don't brush your teeth, often you can start to get these plaque biofilms, which are just bacteria accumulating because there's a lot of food that you ate that you didn't brush off, and there's a lot of carbon sources for them to form, and so that's why you start to see things like this. So they can be bad, and then they can be good in other ways. So what is the extracellular matrix that holds this biofilm together? So in other words, what are the bricks and mortar for biofilms? So what is holding them together? There are two main components. So the biofilms are held together by an extracellular matrix, or ECM, that's composed of two things, Curli fibers and cellulose. So Curli fibers are protein-based. One of the professors at Michigan that I worked with, he studies these in the context of neurodegenerative disease, and I'll talk about that. It's pretty cool. But these Curli fibers basically are protein-based, and they are resistant to denaturants. So they help to keep the biofilms together. But then there's also this other structure of cellulose. So these are glucose fibers. These are also resistant to denaturants, and they're highly wettable. So that's what they look like when you have this extracellular matrix. And so together these form this very tough matrix that's resistant to denaturients. So for these Curli fibers, these are cool because these are actually have been found to be made in our gut bacteria. And these gut bacteria, so they make these Curli fibers and then they form these amyloids. I don't know if you guys are familiar, but in neurodegenerative diseases like Parkinson's, for instance, amyloids are what form in your brain, and that causes your brain to degenerate, and then you start to lose some of those functions. So the functional amyloids that our gut bacteria our gut bacteria basically makes these fibers that create these amyloids, and then that has a connection to seeding the amyloid plaques in the brains that are found in patients suffering from neurodegenerative diseases. So there's a gut-brain connection there with our gut forming these fibers through these Curli fibers, and then that making amyloids that then seed the amyloid fibers that are forming in the brain and breaking down the cells. But that's just an FYI. But just know that the Curli fibers are protein-based, cellulose is glucose-based. Both come together to make this tough extracellular matrix and make it resistant to things that can denature the biofilm or break it apart. Okay, almost done here. So biofilms can serve several functions. They can take different forms and have different functions for different species. When nutrients are plentiful, they can kind of hunker down and wait. They can help bacteria have shelter from harsh environments like antibiotic treatment. And they're important because they help to ward off predators or prevent phagocytosis by cells of your immune system. So they help to protect the bacteria that are encompassing it. And what triggers biofilm formation? So there's different environmental signals and cues like pH, iron concentration, temperature, oxygen, the presence of certain amino acids. So those are just some general things. And then the last slide here is just stages of biofilm development. So you have the initiation stage where you have certain individual planktonic cells. So these are individual cells that will attach to a cell surface, and they coat this surface to which more cells can then attach. So we have these microcolonies. And as more cells bind, they start to communicate with each other, which is that quorum sensing that I talked about. And so once they recognize that they reached a certain cellular density, it triggers this tenacious binding to the surface and to each other through production of this EPS, polysaccharide. And so as the biofilm then starts to mature and form this tower, it makes these channels in which nutrients can flow in and out. And then once nutrients are scarce, which is showing here, then individuals can start to detach from the biofilm and start foraging for new resources or nutrients. Any questions? All right. So the worksheet that I gave you, or I'm going to give you, is going to be helpful in reiterating some of these concepts, and we can definitely talk more about this next week on Monday. But just, yeah, let me know if you have any questions. I hope that was helpful.